produced 6.03 million ton, that is about 355 lakh bales, with a productivity of 480 kilograms. As you know very well that after the adoption of the BT cotton, before that it was about around 300 kilograms of lint per hectare, but at present it is 480 kg per hectare. But even we went up to the, during the this BT era only, it was almost, we reached 560 kilograms per hectare, but it has come down a little bit now. Our target by 2030 is 8.95 million tons. It is almost 50% more what we are producing at present. And by 2040, it is going to be double. The requirement will be double. That is 12.12 million tons of lint is required for the country for home consumption as well as for the export. And double we have to produce. By 2050, 15.29 million tons is the requirement of the cotton. It is going to be 2.5 times. Oh, whether it is going to be possible or not, we have to think of. But there is no going to be any increase in the area. It is going to be just maximum of 12 million hectares for the cotton. So the productivity at present is 480. By 2020, to produce about 50% more, it is required to have the productivity 746 kilograms per hectare. It is going to be 5% more. And like that, 2040, 1010 kilograms. By 2050, it has to be 1274 kilograms per hectare. If you compare with some other countries where they are producing more than even up to the 2000 kilograms, I think it is possible. Only the thing is we have to look into the technologies and also the cultivation aspect, what we have to bring in, especially for the cotton, to meet the requirement of this cotton production. And to name some of the the BT cotton is the one which has made us to double the productivity from 2002 to now. And there are some new technologies like plant genomics, marker assisted breeding, genetic modification, genome editing. These are some of the things. If we are going to look into, I think it is possible to achieve the requirement of the country. Today, our guest speaker, Dr. Samir Sawant, is going to talk on genomics efforts of National Botanical Research Institute for the cotton improvement. And it is a great pleasure to introduce Dr. Samir Sawant. He is presently working as a senior principal scientist in plant molecular biology and genetic engineering division at NBRI Lucknow. He is a fellow of National Academy of Science and fellow of International Society of Environmental Botanists. And he has, at his credit, number of awards, several awards. To name few, he was awarded with Anil Kumar Bose Memorial Award by INSA, Young Scientist, Young Senior Initiative Biotechnologist Award by DBT, INSA Young Scientist Medal, Innovative Young Biotechnologist Award by the DBT, and also he has got CSR Technology Award. Like this, many awards he has got, and he has handled more than 25 research projects funded by CSIR, DBT, ICAR, SERB, and also DST. And some of the significant research achievements, especially in the cotton genomics, he has made. That is the development of large-scale genomic resources, then the novel molecular marker database, SSR and SNPs. And he is the first to develop the first SNP chip for the cotton. Like that, in the number of significant research achievements are also there at his credit. And he has published more than 60 research papers with very high impact factor, even up to the 13.164. And he has also contributed to the books also. And most important is he is having 18 patents. Most of them are of US. So this is very interesting. And also his academic contribution is also not less. He has guided more than 40 PhD students for their research program. He's a member of international and national committees, so and so forth. If I go on reading, it is a very big list of credits. And as you know very well, that I know him very well. He's my good friend, and we interact regarding the cotton research. And now I request Dr. Samir Sawant to deliver his lecture on genomics efforts of NBRI for the cotton improvement. Thank you. Thank you very much. And Dr. Samir Sawant, please.
Yeah, so thank you, Dr. Khadi, for generous introduction of mine. I hope that I will live up to the expectation in my presentation. I thank Dr. Tikku, Dr. Khadi, Dr. Sharan, and Dr. Aparna and also ATPPR for inviting me and giving this opportunity to talk about cotton genomics efforts of NBRI. So I'll just start sharing my slides. Yeah, so my slides are clear to everyone?
Yes, sir, it is.
Yeah. So, we have started working on different aspects, especially genomics of cotton, although cotton has been a mandate plant for National Botanical Research Institute, which is a CSR institute in Lucknow. And presently I am working as a chief scientist in molecular biology and biotechnology division. Our earlier efforts was to develop on I joined this molecular biology division as a JRF with my PhD guide, Dr. Rakesh Tuli, who was also a very well known scientist in the area of plant molecular biology and biotechnology. And I think he's one of the scientists who has actually developed first endogenous synthetic gene for insect resistance. Although my PhD work was majorly confined on gene expression, developing promoters, synthetic promoters, expression systems. In 2006-07, I think we have started, I have shifted my interest from also from transcription regulation and the promoters which I was working to genomics of cotton. So I will be talking like almost last 15 years I've been working on cotton. During this particular period, I have opportunity to interact with some of the renowned scientists in the area of cotton like Dr. Khadi, Dr. Bhattacharya, and I think they have enriched my knowledge about cotton. This is a journey which I would like to present. I don't know about the audience may be a diverse, so a few of my slides may look very advanced for few people, and for some slides will look too basic for some of the experts in the area. So I am sorry for that, but definitely I will be happy to answer questions at the end of my talk. So I structured my presentation by introducing about a cotton, although Dr. Khadi has made my job very easy by introducing cotton in detail. Although I will introduce a little bit about a cotton, then I will go on talking about the genomic resources, how NBRI contributed in developing genomic resources. I will also talk about our efforts on drought tolerance. Then I will also be discussing about gene discoveries in cotton, especially related to fiber traits, and then our recent interest in epigenetics and a novel reversible mesh sterility system which we developed at NBRI. So this is a talk. I hope that I will be able to finish that in 1 hour. In some slides, maybe I will be relatively fast. But if somebody is interested to know technical details about those work, I will be happy to answer after end of my talk. So coming to the cotton, I think there are four important cotton species which are cultivated cotton species. Two of them are allotetraploid and two of them are diploid. So allotetraploid species are the AD genome species. Out of these four species, Gossypium hirsutum is a very important species which actually contributes more than 90% of total cotton production worldwide. Definitely, this is because of a superior fiber quality and fiber yield. Then we also have a Gossypium barbadans, which is very well known for its fineness in cotton fiber quality. But there are other two species, Herbaceum and Arboreum. They are the diploid species, but they are the outstanding source for genes and alleles for various biotic and abiotic stresses, especially on Gossypium herbaceum, where we actually did a lot of work on Gossypium herbaceum during our work. So I will be discussing about our work on Gossypium herbaceum. Coming to the evolutions of this species is very interesting because now most of the species have been sequenced, the genome sequences are available. The polyploidation event is supposed to happen around 1 to 1.6 million year ago between D genome progenitors and A genome progenitor. So D genome progenitors, it is now very well known that for all 8 allotetraploid species which are there in case of Gossypium, they are all AD genomes, and invariably the D genome donor is Gossypium ramondi. The genome sequence of this is very well available. There was a lot of debate whether who among Gossypium herbaceum and Arboreum are the progenitors for this allotetraploid species, whether it is a Gossypium herbaceum or the Gossypium arboreum. Now it is known that since the genome sequences of most of the species are available, it is now known that a speciation event has happened very recently, much before the polyploidation event, which has happened around 1 to 1.6 million year ago. That indicate that this both herbaceum and arboreum has evolved from one a common progenitor that is now considered as a A0. So exact the progenitor for A0 is not still very well known. Now what we have now are some of the wild species of wild form of this particular species, like say for example for Gossypium barbadans we have Peruvian, for Gossypium hirsutum we have Ugandans, for Gossypium herbaceum we have Africanum. For Gossypium arboreum we do not have a wild form. You can see that from both that they are diverse in both their non-fiber tissues in their phenotypic diversity, and you can see that why the AD genome or allotetraploid is very superior, because there's a very higher amount as well as a spinable cotton, and that is why both the species Gossypium hirsutum and Gossypium barbadans are very important as far as the cotton cultivation is concerned, as well as the because in case of worldwide, I think the most of the hybrids come from Gossypium hirsutum and Gossypium barbadans. Now, as far as the genome sequencing effort is concerned, it is the Gossypium ramondi, which is a D genome progenitor, which was sequenced in 2012. Now then actually the D genome was refined by other groups. Gossypium arboreum was sequenced in 2014, and the first AD genome sequence was available, which was sequenced by BGI group, that is the Chinese group. And then after 2015, there are several groups which has improvised this AD genome sequence. Like in 2020, the Wuhan group, again a Chinese group, has a very refined Gossypium hirsutum genome available. This slide is very important, which actually indicate a genome sequencing effort and the genomic resources made available to the cotton community only after 2012. That means before 2012, there are hardly any genomic resources, and that is why the contribution of the NBRI, which I will be speaking to you was very important because NBRI started working on cotton genomics somewhere in 2007 and 8 actually, when there are no resources on cotton genome sequence. There are hardly molecular markers were available, and that made actually the life difficult. But I will tell you, some of the slides look very primitive, but those are very important contribution at that time. So coming to some contribution, I think Dr. Khadi has already introduced cotton, so I need not to go in much detail of this. India ranked first under the cotton cultivation worldwide. India also ranked first in total cotton productions. However, I think this has been already emphasized by Dr. Khadi that India is very poor about yield is concerned. There are several reasons to it, and I will be discussing my next slide what are the major challenges for cotton in India. So cotton is grown in all the three agroclimatic zones, both North Zone, Central Zone, and South Zone. However, the major yield comes from the Central Zone, and Gujarat and Maharashtra contribute a major cotton production in India. So what are the major national challenges in cotton? So like say both biotic stress is a major challenge for cotton. Both insect and pest are the major cause. Both chewing and sucking pest are the major cause. The chewing and sucking pest actually, although I will not be discussing, although NBRI do a lot of work on this particular thing, although that work has been being conducted by my another senior colleague, Dr. P.K. Singh. I will not talking about that particular work, but this is a major challenge, both chewing and sucking pest, for the which actually are responsible for significant loss in cotton. We handle abiotic stress tolerance. Drought is another major problem for cotton, which affect both cotton physiology as well as cotton productivity. Drought is important because still more than 65% of the cotton agriculture is in red condition. So actually you can see that with the climate change and with unequal and erratic rain pattern, actually the drought has become a very significant problem. So developing a drought tolerant varieties are one of the major challenge for cotton agriculture. So need to develop a high yield and resilient cotton varieties, and we require now innovative technologies. I think Dr. Khadi spoke about improving yield. So say high density planting actually is one of the things which actually all the cotton breeders or the cotton scientists they are considering can have an impact on improving yield in cotton. So we require technologies for developing varieties actually which can be grown in high density planting, and also the machine picking which is very much required because cotton agriculture has become absolutely very expensive, and innovative technologies like machine pickings are required. So these are the major challenges, and we have to develop varieties for each of these particular challenges. So with this introduction, now I come to what exactly NBRI do. So NBRI is a major group working on pest control. I think NBRI has contributed 2 major genes, Cry1 EC, and then now recently I think there was a major success story of TMA12, which is for whitefly resistant, which has been developed by my colleague Dr. P.K. Singh. Then we work on drought tolerance. We also work on development of markers and several traits in cotton, including yield and quality. So I will be speaking majorly on drought tolerance and our efforts on yield and quality. Now, so this is the first topic of genomic resources development in cotton. So our work started with, say I said that Gossypium herbaceum, like our major interest initially was developing drought tolerant varieties. So we focused on Gossypium herbaceum because this A genome are A genome species, both Gossypium herbaceum and Gossypium arboreum. They are very tolerant to the drought. So the idea was to identify both genes, alleles, and markers from this, and making use of these genes in Hirsutum to develop the drought tolerant variety. So when we started, there was hardly any SSR's information, so marker information available in cotton microsatellite database, especially for Gossypium for the age, So we decided to enrich cotton microsatellite databases, me and one of my colleague, Dr. Jena. And, we started developing SSR enriched library, and this SSR enriched library, initial sequencing of this SSR enriched library resulted in development of some 263 genomic SSRs. This numbers now looks very small, but that time actually when we contributed in 2012, it was a major contribution. We were also doing a lot of transcriptome sequencing on that is RNA sequencing in Gossypium herbaceum. I will be speaking about that in second part of my talk. So that EST sequences also we screened for a lot of SSRs that has contributed to almost 2000 express SSRs. These SSRs, both genomic SSRs and the expressed SSRs, we have actually used for genotyping several Gossypium herbaceum accession which were available to us. Actually, this accession made available to us. We had a collaboration with US Harvard that time, as well as JK Agrigenetics. So they were the major contributor for donating this herbaceum accession to NBRF. So we could group, we could develop this, good tree. We can actually do a lot of basic studies on this Gossypium herbaceum using this SSR. We could also see that the transferability of Gossypium, the markers, both the genomic SSR in ESSRs which we developed was very good from herbaceum to arboreum to hirsutum to barbadans, and that means these markers can be also being used, which has been developed in herbaceum or in arboreum as well as in hirsutum and in barbadans. So this was our earlier contributions of developing genomic SSRs and express SSR, which was published in Theoretical and Applied Genetics in 2012. Now coming to our large-scale genomic resources development, so this is actually come with our collaboration with JK Agrigenetics. So JK Agrigenetics, that time actually we connected with JK Agrigenetics under one CSIR program which is called the NIMITLY program. So that is a CSIR program which actually initiated a collaboration between a CSIR and a non-CSIR academic institute with that of industry. So under this this program, we were supposed to develop a lot of markers for both for Gossypium hirsutum, because this program also started in somewhere in 2008 and 9. That time, no genomic resources, and the idea was to develop a dense linkage map which can be used for for QTL identifications for both fiber quality and for yield. So that was a major challenge that time. So we use a technique called HMPR. So HMPR stands for hypomethylated partial restriction. This technique is widely used that time for actually making a genic enriched library. So I'll just explain about this particular technique. So this technique makes use of restriction enzymes, which are a methylation sensitive restriction enzyme. So like for example, we use two restriction enzymes here. One is HPA2 and CLA1, both are methyl sensitive enzyme. That means if the particular sequence is methylated, this restriction enzymes cannot cut that to those sequences. Unlike those enzymes, like say restriction enzymes like EcoR5, N3, these are methylation insensitive enzymes. That means invariably, whether it is a methylated or unmethylated DNA, it can cut a genomic DNA. So like say for example, if you cut with HIN3 and EcoR5, you get a uniform smearing of genomic DNA in the gel. But if you cut with a restriction, if you cut a genomic DNA with the restriction, this methylation sensitive restriction enzymes like HPA2 and CLA1, it will do a partial restriction. This is because the uncut DNA. So the idea, because say it is very well known that the genic regions are generally hypomethylated regions, and the regions which are near centromeric or peroxymetic regions which are highly condensed, they are highly methylated regions. So definitely we were interested in developing markers which are enriched in the genic region. So this technique was used to develop genic enriched library. So we use this particular technique for developing genic enriched library. So that time actually we got several genotypes from JK Agri Genetic. Some of the genotypes has been classified as good for fiber quality. I will be explaining that in some of my lectures, right? So we took total 6 genotypes, and all these 6 genotypes were digested. We developed this both libraries for using this methylation sensitive enzymes HPA2 and CLA1, and we did this sequencing. So during that time only we got like 454. It was probably the first installation of 454 in India, which is a pyrosequencing technology. Now, if you look at the amount of data which is being generated, it looks very small, but that time actually when we're doing in some 2010, 2012, actually, the per run, the sequencing data actually could generate in case of 454 was almost 250 megabits per to 500 megabits per data could be generated in per run. Like now we have technologies which are available which can go in terabytes of sequences. Like say for example, in case of Illumina NovaSeq 6000, you can generate more than 6 terabytes of data per run. So this amount of data that time, so total like say around 5.2 GB data was generated for all these 5 genotypes, and this data was we have used for developing markers. So I will be explaining it to you. So like this is a strategy. So like it was very important because say we were doing this sequencing in 2010-11 where there was no reference genome was available. Assembling now the sequences when the reference genome is available is very easy, but de novo assembly of 5 GB data was that time technically very, very difficult actually. So, we have to develop our pipeline. Thanks to my colleague, I will be talking about their work also, to my both colleague, Dr. Meher Asif and Dr. Sumit Bagh, both of them, help us in developing, bioinformatics pipeline, which help us in assembling this particular data. You can see that, we could assemble this particular data, and the quality of this, this Q40 scores is very good, more than 90%, indicating a very good quality score. Now this assembled data was used for the marker discovery. So our interest that time was developing both SSR markers as well as SNP markers. So we use this particular, this particular pipeline, we have developed, this is an endogenous pipeline for developing SNPs, identifying SNPs, because that time the genome sequences of both, even the D genome sequences was not available. I will tell you that a little bit later. So assembling, de novo assembling was a challenge, but we could do that. We assembled the sequences. This actually the assembly leading discovery of a large number of SNPs, probably the largest that time actually when we were doing this, So initially we could identify something like more than 4 lakh SNPs, but after filtrations, after a lot of quality control, we could reduce from 4 lakh to something like 75,000 SNPs. Since 75,000 SNPs, when we removed the redundancies, we eventually got something like 66,000 SNPs, which are completely novel. So this is a novel SNPs, like they were not reported by anybody, any group. This was the first time that we were contributing the 66,000 SNPs. Out of the 66,000 SNPs, most of these SNPs are from genic and non-genic area. Non-genic area are generally confined to the near gene region, that means the regulatory regions. And also we have a lot of SNPs in both exonic as well as in intron and 5' UTR region. So this SNPs, the exonic SNPs is been then further classified into synonymous SNPs and not synonymous SNPs. So these SNPs, some of these SNPs are further then validated by sequencing those, because it is very important once you identify those SNPs, we need to show, because genome sequence was not available. The major challenge was discriminating SNPs from homolog, because Gossypium hirsutum is a allotetraploid having 2 genome, A and D genome. So, classifying 2 SNPs from actually, homolog was a challenge that time. So some of these SNPs were actually validated by doing the sequencing around the SNP regions, and many of these SNPs were validated. This validated SNPs I will be talking later has been used for the purposes. Since like large amount of data was also available, my colleague Dr. Jaina made use of this data for identifying SSRs. Large number of SSRs which we could identify. So there are a lot of quality control which we did, which actually resulted in identification of something like 47,000 novel SSR. I think altogether that time actually when we actually deposited this 47,000 SSR, the CMD database, that is the Cotton Microsatellite database worldwide, were having something like 24,000 SSR when 2011-12 when we deposited this. So there was a large contributions which we did. We also sequenced, because we also sequence some of this SSR region and we show that this SSR are the true SSR. So this is the total contributions of different project which actually did in a significant marker development from NBRI. So like under different project, like say as I said that from sequencing various genome SSR enriched library as well as transcriptome, we could develop more than 2,000 SSR. But the major SSR discovery come from our this HMPR or the Genic Enriched Library, which contribute something like 56,000 SSR and 66,000. So total 129,000 markers together we contributed. This markers, like if we, I think in 2012, this 2013, when this D genome was made available, we actually mapped this SSR and SNPs onto the D genome. Now this is only D genome. Okay, that's a random Ramonde genome, because AD genome was not available that time. And you can see that this SNPs and SSRs was uniformly distributed across all the chromosomes, indicating their utility for developing the good map for Hirsutum. So our work, which was then actually published in 2013, which was a little bit little bit later than the D genome was made available. So it was considered by Plant Biotechnology Journal as one of the significant contribution in the area of cotton genomics, which they have actually projected on their cover page. So this is all together we have contributed, because the AD genome sequence was not that time available, and all these resources came from Gossypium Hirsutum. Now this is actually, I was talking about the work. So most of the bioinformatics work was done by Dr. Sumit Bak and the students which are working with Dr. Sumit Bak and Dr. Meher Asif. This actually did in development of this comprehensive cotton genomic database. So whatever now gene discoveries, SNP discoveries, or the genome sequencing which we are doing, we keeps on depositing this data to this particular database, so that every time actually we keeps on this database updated. This database is now been widely being used by many people. So I just give you an example that SSR, which has been developed by NBRF, people are making use of these SSRs, and some of these SSR are being projected in their QTLs, actually, the SSRs which NBR has identified. So like say the another target was to then develop SNP chip, because Affymetrix then contacted, because we were the major contributor for cotton SNP chip. So Affymetrix then contacted us to develop the chip, and we could develop 50K cotton SNP chip. That was the first cotton SNP chip developed by us, and which has almost SNPs coming from 42,000 gene, and for this 42, it has actually 51,000 probes, which covers almost 42,000 SNPs. Out of this 42,000 SNPs, most of the SNPs are actually from our in-house sequencing effort, and some of the SNPs which are also included from the public to validate our actually the SNPs as well as our chip, actually, So this chip was available to us for a lot of other work which we did with this cotton SNP chip. So the first the project which was sanctioned by DBT, that is in collaboration with CICR Nagpur. So Dr. Wagmare, actually we collaborated with Dr. Wagmare with this 50K cotton SNP chip. Dr. Wagmare that time developed one H into H population, and we genotype this H into H population within the 50K cotton SNP chip. And if you can see, a very dense, a very high density linkage map which we could develop actually. And the number of, you can see the average distance per marker is almost 1.31 centimorcan, which is very good actually for the cotton. So this particular linkage map we have also used for identifying this SNPs for several traits in cotton, which includes fiber quality trait as well as field trait. And some of these now QTLs are under validation with CICR Nagpur in collaboration with Dr. Wagmare. So looking at our efforts with this, actually DBT has also then sanctioned a national program on cotton actually. So where actually we were supposed to genotype a total 6 populations actually. Two populations from CICR Nagpur. So one population was H into H, another population is H into E, A, and then we have also collaboration with US Dharwad, where we are actually using this 50K cotton SNP chip for genotyping H into B population. Then also this they also US Dharwad also has a core collection, so we are genotyping that also using this 50K cotton SNP chip. We are also having a collaboration with TNA Coimbatore. They have developed 2 H into H populations, and we are doing genotyping of both these H into H populations using this 50K. We initially IRI was also part because they have one H into H population, but since like Dr. Bhatt, who has developed this particular population, he then eventually retired, and then this population was transferred to US Dharwad. So actually this population has been now taken care by US Dharwad. So we are now almost completed thanks to my colleague Dr. Jena. We have now completed all the genotyping of all these populations, and presently so you can see that this all population, we have the a good linkage map for all these populations which are available. Some of the linkage map was not as expected good, mainly because they are either they are H into B, we are facing some problems, so we are improvising on that actually. So we are now making this linkage map for QTL mapping, and very soon I think our work on QTL mapping will also get completed with all this population. This project was done with the support of DBT. Dr. Jena has also made another approach actually, because there are a lot of QTLs for both fiber yield and for quality is available in public domain. So he actually developed a consensus QTL map for all the QTLs which were available in the public domain, and he could identify 2 major QTL hotspots. Basically, his interest is ball weight, actually, which is one of the very important agronomical trait in cotton. So he identified 2 major QTLs, one on chromosome D6 and another on chromosome D4. And presently, he is actually these are the 2 hotspots. And presently, he's targeting. He's doing a targeted sequencing in both those regions to develop a markers. So probably we will be coming out with a very specific markers for the ball weight. We made use of this particular chip. We also Nuzividu also collaborated with us for this 50K cotton SNP chip. They had interrogation lines from Gossypium iridium. Their interest is their interest was in JACID resistant, and Gossypium iridium was a JACID resistant donor. So they have used this introgress lines, that is 7076 and 7082, and they have used 2 recipient parent BS1 and GP122, and they developed 11 near isogenic lines. one under one under the background of 11 under the background of BS1 and 5 under the background of GP122. So we did the genotyping for all these near isogenic lines as well as their parents, and what we could identify that could identify that 200 SNPs are commonly introgressed from these donors into this 11 NIS which we actually genotyped. Now, interestingly, that out of this 200 SNPs, 32 SNPs was actually clustered into one particular hotspots of 18 megabase per region on chromosome 8, that is DT8. So that indicate that probably the GESID resistance is linked to this DT8 chromosomes. And actually we were that time discussing about doing either targeted sequencing or the fine mapping of the 18 megabase per region. Somehow this project was halted here, and actually we could not take it further, this particular result. But I think we could tell them that actually this with our this 50K cotton SNP chip, this 18 megabase per region is actually responsible, which has been introgressed from iridium and actually responsible for GESID resistance. Okay, so with our effort which we are doing on the marker development, the international collaborations actually which has been that time actually been steered by Dr. David Stille from Texas Agricultural University, Texas, and then he contacted us to become a part in another cotton, global cotton SNP chip, which was getting developed with Illumina under the Illumina background, where the NBRI contributed SNP there also. And so like NBRI contributed SNPs both in one is like our endogenous cotton SNP chip which we developed, which are majorly the SNP coming from the genotypes which are the Indian Gossypium hirsutum genotype, which are contributed by AgriGenetics, and also some of our SNPs which were developed eventually become a part in cotton SNP chip developed by the international consortia. Now this is a 63 cotton SNP array which is available worldwide, and it is now the most commonly used cotton SNP, because this is having SNPs not only from hirsutum but many other species. As you can see, it has a SNPs from barbadans, other adigenome species, Mesotellinium, then Gossypium longicaulis, Armonarium, Tomentosum, so several species actually. So this particular chip is very popular, and worldwide it has been used for several genome-wide association study. Now coming to our work on drought tolerance. So as I told you that our initial focus on Gossypium herbaceum, because they are known to be tolerant. So we screen a lot of germplasm of Gossypium herbaceum with several methods. I think the initial methods is like screening with mannitol. Eventually, what are the tolerant varieties or sensitive variety which we got? We did some pot experiment, then field experiment, and then we actually identified two genotypes. The one Vagat variety genotype, which is a Gossypium herbaceum genotype, and this has been identified as the most tolerant. And another genotype, RHS14, I think this has been contributed by US Dharwad to us, and this has identified as the most sensitive genotype. We did the detailed physiological studies of these two genotype, and we identified that Vagat, which I understand has been also grown in several almost deserted area in Gujarat actually, where there is a significant high salinity and drought tolerance is there. This has actually evolved with this drought tolerance stage, and when actually we did a lot of physiological parameters, what we identified is drought tolerance in Vagat is mainly because of its immediately as soon as it faced drought, it actually reduces its tomatal conductance, it decreases its transmissional rate, it reduces its water potential, it increases its higher what that actually this result into higher relative water content and eventually better water use efficiency. So this was actually the Vagat was a very good genotype for identifying genes and alleles for drought tolerance. So we did a lot of transcriptome sequencing. I will not be going about technical details about transcriptome sequencing. And then this data is been analyzed. Eventually, what we identified that this WAGAD has a several pathways which are known to be involved in drought as well as abiotic stress called lectophenide pathways, and several genes which are responsible for abiotic stresses or the secondary metabolites. These are quite enriched in this WAGAD varieties when the drought stress is given to this, and these genes becomes the resources for us for gene mining and then using these genes and alleles. I will be giving some examples of this. And RHS14 is a sensitive variety. As soon as it suffers drought, actually a lot of ethylene responsive pathway get induced, which results into programmed cell death, senescence. So this is what actually happens. So that actually answers that why WAGAD is a tolerant genotype and RHS14 is a sensitive genotype. We also did a detailed sequencing of from the roots of it, and could identify potential genes which can increase the root biomass, because it is expected that genotypes which are deep rooted or having a better root biomass will actually sustain abiotic stress condition much better, especially the drought condition. So the idea was to identify the genes. So actually with this sequencing resulted in identification of the two important genes which presently being carried out, which we are actually doing in detailed studies. So as you can see that one of the genes which belong to a family called CAMTA, that is a calmodulin activated activator. So this particular transcription factor which specifically express, so TR indicate tolerant root, and that is a WAGAD root. And see, you can see that it has a very high TPM in the root, but it is almost absent from sensitive leaf as well as sensitive root, indicating that this is specifically expressed. So when we validated this particular gene using a real-time PCR, we see indeed that this particular gene is actually expressed in the root. So we decided to focus on this particular gene. We because say cotton transformation still a challenge. So initially we validated this gene in case of tobacco. So several tobacco lines were developed overexpressing this particular gene, and you can see there is a significant increase in the root biomass when we overexpress this particular gene. Not only root biomass, overall biomass, and the plants were quite tolerant to the drought condition, and you can see from carbon discrimination ratio also. So carbon discrimination ratio is inversely proportional to the water use efficiency. That means the lower is the carbon discrimination ratio, better is the water use efficiency. So you can see the overexpressed lines of cotton, this CAMTA in case of tobacco, as a much lower carbon discrimination ratio that indicate higher water use efficiency. This plant, since they have a better root biomass, we also see that they have better nutrient utilization efficiency. I won't be going detail about this because the detailed work is presently going on. Now we also develop some primary transgenic lines overexpressing this particular gene in cotton, and you can see the biomass. These all lines have been developed in cocker because that is the only genotype which is actually which we can use for genetic transformation. So you can see that there's a significant higher biomass of the in a line, the transgenic lines which are overexpressing this. So likewise, in case of tobacco, they have a better carbon discrimination ratio. So like more lines are presently getting developed in my laboratory, and this will be then evaluated. And if we found that a plant, then we will go to field trial of this overexpressing lines. And if we found this usable, then I think we will transfer this by using a backcross strategy. This genes will be transferred to Gossypium hirsutum, the varieties which are popular varieties of India. My another colleague, Dr. Vidhu Sane, also identified another transcription factor which is called a NAC transcription factors from again the same WAGAD variety, and actually she also identified the same gene specifically expressing root. And as you can see that when the genes were expressed in cotton, they have better root biomass, and they are the deep rooted biomass. And the transgenic line she has also evaluated the overall yield seed yield as well as the fiber yield, and she saw that the plants which are the transgenic lines overexpressing this gene are better in both drought response as well as in so like presently under a CSR project we are now actually making use of this both NAC gene and CAMTA gene. We are doing a targeted sequencing across different varieties, and the one idea is to the one approach is transgenic approach, another approach is developing marker for this NAC gene as well as for CAMTA gene, and using this marker for then actually molecular assisted breeding for improvement of cotton. Another colleague actually, since like we were doing a lot of studies on Gossypium herbaceum and we identified that herbaceum, this especially Vagard variety, is a very good source for allele for drought tolerance. So, you know, Dr. Hemant Yadav developed tilling populations using the EMS mutagenesis. So I won't be going in technical details of this. Like, you know, so we have something like more than 6,500 tilling populations. This 6,000, you know, you can see the phenotypic diversity of the tilling population which we developed. Since like we were very keen on drought, so what we did some field trials of 2,000 of these tilling lines, and in both irrigated and you know control drought condition. And what we identified 42 mutant lines which are tolerant than the parent Vagard, and there are 104 lines which are sensitive. Now this become a very good resources for identifying genes. So under a CSR project, like now we are actually selected some of the most potent line, that most tolerant and most sensitive line, and we are using a mute map approach, you know, to identifying actually, you know, these genes actually which are probably this will give one further new genes for drought tolerance from this Gossypium herbaceum. Definitely, so we required for mute map approach because we will be doing a F2 population sequencing, that is a bulk segregant population sequencing. So we required a good genome of Vagard. So we completed recently the Vagard genome sequencing. We have now Oxford Nanopore Prometheon genome sequencer, which is a long read sequencer. So we made use of long read sequencing as well as small read sequencing to sequence this Gossypium herbaceum Vagard variety, and we have a reference genome. So once we get a F2 population, it will not be difficult to map those and to identify those genes or the and where the mutations has happened in those, and probably those will give a better you know both genes and markers for to be used actually in our future study. Now I will quickly go to the work which we are doing in cotton yield and quality. This is, I think, our collaboration with Tiara, which we still continuing. So our interest with Tiara is actually developing marker for high density planting and machine picking traits in Tiara. So we have now two different populations. So initially our population was confined to 219 lines in cotton, and we have considered 15 traits, and we use 30K SNPs using a GBS sequencing, which is a restricted kind of approach we use for this. That did is identification of several you know SNPs, which are what we call as MTA, that is the marker trade association. So this MTAs, when we classify this MTAs, like we have these markers, and you know some of these markers are presently under validation in a biparental populations. Especially we are very hopeful for some of these markers which are developed for ball weight actually, you know. So because in various statistical studies, this actually ball weight emerged out as the potential markers actually. So we will be focusing on that. So Tiara has also contributed under a collaborative research 1,000 genotypes they are donated to NBRF. Out of these 1,000 genotypes, like we have developed now 320 core, 320 line core. Now this 320 core, like we are now actually considering several agronomically, economically important traits in cottons, and presently the multi-location trials are going on in Aurangabad, Yavatmal, Hyderabad for both drought tolerance as well as for various, and I think we will be sequencing, in fact the genome sequencing of these 320 lines are presently going on, And this genome sequencing earlier approach was a more limited approach with GPS, that is a more restricted approach. Like here we are doing a entire resequencing of this 320 germplasm. So we expected to get a very important alleles and markers for various agronomical traits. So coming to the gene discoveries and their validation. So I think for people who are not from the cotton background, this cottons are a single cell trichomes, actually which actually originated from the epidermis of the ovules when the fertilization happens. So the day the fertilization happens is called first day, 0 day, we call it 0 day anthesis. And actually the cotton initiation begins just 1 day before the anthesis. So it has been the entire development of cotton can be actually been grouped into initiation, elongation, secondary cell wall, biosynthesis, maturation. These are all overlapping stage, but there are distinct stage. There are different group of genes which actually express. I will be explaining that to you. So our initial work was on the genotype which were contributed by JK Agrigenetics. So two genotypes, JKC725 and JKC777, which was contributed as a superior genotypes. And there are three genotypes, that is 703, 737 and 783 was contributed as an inferior genotype. So you can see that why they are superior by looking at their fiber quality. We did some studies and also found that that these are actually superior in their fiber length, and you can see the lignin content and cellulose content, which actually reflect the superiority of 725 and 777 over the 703, 737 and 783. What we did, so this is another very significant study which we did, that we took these 6 genotypes and 5 developmental stages like 0 DPA, 6 DPA, 9 DPA, 12 DPA, 19 DPA and 25 DPA. So these 5 DPA represent both initiation, elongation and secondary cell wall biosynthesis. And so 6 multiplied by 30 and then 3 biological replicate for each of this stage. So total 90 micro array studies which we did, because this study that time the entire when we were actually doing this work in somewhere in 2010-11, transcriptome sequencing was was just emerging out, and a cotton a very good cotton array was available, expression array was available with Affymetrix. So we made use of this Affymetrix cotton expression array, and we could identify interestingly genes which are either DPA specific, that is the development specific, or germplasm specific, or interaction specific actually. And you can see that these genes can be classified. There are specific group of genes which are expressed at different levels, like say these particular clusters can be grouped into initiation, like cluster 1 and 4, they are the initiation specific cluster, cluster 2 and 5, they are elongation specific clusters, cluster 3 and 6 are the secondary cell wall. And they are the distinct clusters having got different genes. Actually, they have given us an idea that what could be the genes which are responsible for the superior fiber quality and inferior fiber quality. I will explain to you. So this has been mapped into a comprehensive map actually, which actually tell us what are the transcription factors, what are the different hormones, what stage of different development these hormones and these transcription factors are working, and some of these genes, some of these transcription factors are presently under validation. So one of this transcription factor is MYB1, which we identified. So like when we're doing this particular study, we identify one particular gene, that is a GDSL specific lipases, and this particular gene expresses specifically during the secondary cell wall. So we were focusing on this and we characterize this particular gene. As you can see that we clone this promoter of this GDSL specific gene and fuse it with a Gus reporter gene. We develop a transgenic line, and you can see that this gene expresses specifically around 19 dpa. Like its expression was high at 19 dpa, which is a secondary cell wall synthesis, and very important stage which actually contribute to the fiber strength and fineness of the fiber. So our interest was to study both the importance of this GDSL gene and what transcription factor actually regulate this secondary cell. So this act as a marker, GDSL gene acts as a marker for the secondary cell wall biosynthesis stage, So we did a detailed dissections of this particular gene, and what we identified that this particular transcription factor, actually when we mutate a specific element, that is a MIB180 cis regulatory element, you can see that expression is completely lost, okay, indicating that the expression of this 19-DP specific GDSL has been regulated by a transcription factor for MIB180. So what we did, that we did a East-1 hybrid interactions with this cis regulatory element, and we identified a transcription factor which we call as a MIB1 transcription factor, and this MIB1 specifically interact with this, and this MIB1 eventually, with a lot of study which we identify, this MIB1 actually controls a lot of genes which actually express during the secondary cell wall state. So presently we are validating the importance of so I think this work is published again in Plant Biotechnology Journal in 2017, but this work is still continuing, that we are actually now studying that how the MIB1 controls secondary cell wall synthesis stage in case of cotton. This is another very recent work which we have recently published, I think in this month only. My another student, Preeti, actually, we have developed a comprehensive, so there is a, like now if you look at cotton expression database, like it has been enriched with lot of RNA-seq data which has been deposited not only by our group but worldwide actually. But there was no effort which can take account of all this data and come out with the expression atlas of cotton fibers actually. So my student Preeti Prasad, actually, she actually compiled all this data, and as you can see that the total data which compiles, which actually covered 400x coverage of AD genome, which is a significant, that means whatever the genes which will now identify from this will be very significant gene, and which actually she has identified. She classified those genes into different developmental stages, and in collaboration with Tiara, we actually validated the genes. So what we did that across several lines now presently under evaluation, we actually did the expressions of the genes which Preeti has identified, and we use a technology called Encounter technology, and this Encounter technology can actually help you in doing a lot of expressions of say several genes at a time in several samples. So like say for example, we did the expression studies in 100 cotton genotypes and 3 developmental stages. So 0 DPA represent initiation, 6 DPA represent elongation, and 19 DPA represent, and eventually we could see that say many of these genes whose expression profiles are strongly correlated with the fiber quality, and now these genes are very important for developing markers, and some of the genes we will be taking in our future study for validating those genes, their importance in fiber quality. Many of the genes which she has contributed are the novel genes, which will be doing it their further validation. So actually, she developed another now database, which also part of our previous database, which we call as NBRS Cotton Expression Database. So this is also available online. Like now, I think I will briefly, because I have maybe not more than 10 minutes left, so I'll briefly go with the epigenetics. So I think we were interested that how, beside genetic factors, how the epigenetics controls the fiber development. So epigenetics can be because of the many reasons like DNA modification, histone modification. So my laboratory's major interest was histone modification. So one of the histone modifications which we are working on, histone acetyl, so histone, this histone acetylations of the histone is being controlled by two different types of histone modification machinery. One is histone acetyl transferases and another is histone deacetylase. So what histone acetyl transferase does, it transfers acetyl moiety from S adenosine methionine. So it transfers the acetyl moiety to histones and acetylate. So when acetylation moiety is transferred, the histones get decondensed and the transcription happens. And the histone deacetylate will deacetylate. That means it removes the acetylation moiety from the histones and condenses the histones further. So So we were using an inhibitor. So there are specific inhibitors known. So one is anacardic acid, which specifically inhibit histone acetyl transferase, and there's another inhibitor called trichostatin. So we decided to study this histone acetyl transferase using this anacardic acid and trichostatin. What we identified that when we treated the ovules, so like the entire cotton fiber development can be mimicked in vitro condition. So there are specific way by which you can completely see the fiber development which is happening in your Petri plate. So we make use of those in vitro assays, and we have seen when we give a treatment to the anacardic acid, we see the significant increase in the fiber biomass. And when the TSA has been treated, there is a complete inhibition of the fiber development actually. So indicate that histone acetylation plays a very important role in case of fiber development. We also seen in case of anacardic acid treatment, initiation happens much earlier. So the interest was immediately to see that whether this technology, anacardic acid, can, if we spray it onto the plant, can increase the yield in cotton. So again, this is at later fiber development stage. So we collaborate again. This technology was developed in collaboration with Tiara Seed Sciences, and we have seen that when we spray it with this anacardic acid, we see that increase in ball weight in case of different accessions of Tiara, and we did some trials, several year trials with Tiara, and we also see early maturity when the anacardic acids sprayed. We also did some trials in Kharif of 2017 in different regions, and presently this technology under evaluation with AICRP, with CICR Nagpur is evaluating. We also did a toxicity trials with anacardic acid, and it is found to be safe with whatever the parameters. We collaborated with UCP Chemical, which are now the bulk producers of this anacardic acid. So anacardic acid, so our basic science has resulted into a technology. So these are the key points of our technology. Actually, we identified that it can increase almost 10% of the cotton yield, and we also see that the earliness is in cotton, which will be important for northern state like Punjab and Haryana. We not only stopped here, we now are trying to identify that what are the genes which are actually regulating this. So one of the genes, one of my student Virendra Kumar has characterized is histone deacetylases. So what we identified, we actually clone this particular gene from this Gossypium hirsutum and characterize that it's actually involved in acetylation of a histone moiety called H3K9. And when we suppress this particular gene in case of cotton, actually it resulted into significant reduction in the ball weight as well as the fiber yield, actually indicating is a very important gene for the fiber yield. So we have, with his study, we could now know that, probably that there is a very big complex which actually governs this entire histone acetylation in case of fiber development in cotton. His work is published in Plant Journal in 2018. We are now actually further continuing characterizing this entire complex in my lab. Both HAT as well as HDAC and some histone methyl transferases are getting characterized in my lab. We are also involved in developing a reversible mel stability system in cotton. So I don't have to tell why hybrids are very important. You can see, although this data is relatively old data, but you can see in 2016-17, 5 crores of H1 hybrids of cottons have been sold in India, and very few companies have a license for genic mel sterility, which has been developed by Monsanto. So alternative systems are needed for developing hybrid. So we have been working on developing reversible mel sterility systems since 2010. We have published several papers which shows, because during the pollen development, degradation of the tapetum at a specific time is very important, because tapetum is a nutritionally enriched tissue which actually provide nutrition. So there is a programmed cell death. So if you disturb this programmed cell death with a gene called Beclin-1, which is basically induces the autophagy in case of plants. So what we did, that we express, when we overexpress this Beclin-1 gene, specifically in the tapetum, plant become mel sterile. So the idea was to do this in a controllable manner. So what we did, that we develop a reversible expression systems where promoters brought under the control by a specific mutation called TGTM mutation. This TGTM mutation, this promoter, that means if you have a TGTM mutation, the promoter will render inactive. But if you made available this TBP mutant 3, which specifically bind to this, the promoter will be active. So, so if we develop this particular systems and we overexpress this particular Becklin gene, the plant become malsterile. Okay, and we fuse this particular TBP mutant 3 with another protein called HFR1, because this HFR protein has been targeted by one ubiquitin ligase, which is and a protease which is called COP1 proteases. So this COP1 proteases, so what we are doing, we are expressing COP1 proteases in male parent, and we are expressing this particular construct in female parent. And eventually when we cross it, so like say for example, when COP1 is not available, this TBP mutant 3 will control expression of Becklin gene. When the Becklin gene will induce, we will get a malsterility. So this has been shown here. So when we have the express this particular construct in first initially in case of tobacco for the proof of concept, and we see that in case of when we overexpress it, plant become completely malsterile. You can see that the control pollens are green in colors, which are fertile pollens, and which is FDA staining. But is the dead pollen do not take FDA, and these have been counter stained. And you see that they have been stained with a red color actually, and the pollens you can see in case of wild type plants. You have the very nicely grown pollens which germinate also nicely, but in case of malsterile plants, you have a completely abnormal pollens which do not germinate, and there is no seed setting happens. So this technology initially we have shown in tobacco. Now we recently introducing, my colleague Dr. PC Verma actually is helping us in introducing this in cotton. My student Rishi Verma actually is a student common to both me and Praveen, is now transferred this technology to cotton, and we see that the technology which was working in case of tobacco also work very nicely in case of cotton. You can see the cotton line, the female lines are completely malsterile with abnormal flower, but when this is crossed with the male line, the fertility is again restored. So we have now the system available. This is a one picture, a very good picture which shows this is the control or the male plant, which is showing a very good, very nicely developing pollen, and female lines. These are typical female lines with no development of pollens or no germinating pollens actually. So we are developing these technologies. This is actually and now actually we have evaluated now several male and female lines in our greenhouses. We have identified potential lines, and I think this technology will be useful in days to come. I think this is a completely endogenous developed technology for developing sterile lines in case of cotton. So I think I know that I did not explain this in technical detail because of short of time. So this is all about actually what we are doing. I have not at all talked about pest resistance work, which my colleague Dr. P.K. Singh is doing. So, but NBRI is contributing in genes for pest resistant, drought tolerance, yield, and quality. We are also developing a lot of markers. We are developing F1 IVD technology. We are working on green molecules like anacardic acid. So together, our contribute for improve cotton yield. So these are the acknowledgement, different scientists, different students. Definitely, I would like to thank my industrial partners like JK Agrigenetics and Nuziviru Seeds, Tiara Seed Sciences, with whom I am doing now recently a lot of, and my government, partners like TNAU, US Harvard, CIC, and Nagpur. And definitely, I thank Nimitli, DBT for giving my CSR and DBT for giving funding support. This is my group. And thank you very much. I will be happy to take now questions.
Thank you.
Thank you, sir. Thank you very much.
Yeah, thank you. Thank you very much, Dr. Samir. And please raise the hand for the any queries or the questions, please, from Dr. Samir.
So there is
One,
Mr. Praveen Prakash.
Praveen Prakash has raised the hand. Please, Praveen, please, you can ask what your queries you have at your end. Praveen, please
Is he there?
unmute yourself.
Is he there?
Yeah, he's there. Praveen, unmute yourself and.
I think
I don't see.
Anyway, one more was there, Dr. Bhima Reddy. Earlier, he raised a hand right from the beginning, but I don't know whether he's there or not. No. Anybody else? Please raise the hand.
Yeah, I would like to first of all thank Dr. Samir.
Yeah. Yeah.
I think there is this Rahul.
Okay.
Yeah, I think he want to ask a question maybe.
Yeah. Yeah, sir. Hello, this is Rahul Fude, sir.
Okay. Yeah.
Yeah,
Okay.
yeah, I am a scientist at CICR Nagpur.
You are?
Scientist at CICR.
Okay. Okay. Yeah, Rahul. Yeah. Please.
So regarding this SNP genotyping chips,
Yes.
we have the magic populations developed with the indigenous parents, like 8 parents, 10 parent magic population. These are having some segregating generation. In future, if you want to use the SNP for mapping those with those magic population, which one will be better, 50K or 63K, as the parents are indigenous?
Yeah, so I think your question is whether you should go genotyping using a SNP chip or you should go for an NGS-based approach. Okay.
Yeah,
So
yeah.
it all depends upon I would say that it all depends upon your funding availability. Okay, because definitely say how many lines, say like for example, if you have 300, 400 lines, say like for example, we are doing now a resequencing of 320 core population of which we developed in collaboration with Tiara Agrotech or Tiara Seed Sciences. And actually it is costing us for resequencing something like ₹1 crore, okay, for resequencing. Even if you go for a genome, but that will be a more comprehensive approach because there you do not limit, say cotton SNP chips actually limits you to the SNPs which are already identified. So, but genome sequencing approach do not limit you to a particular approach. So if your funding supports you, then I would definitely say that if you have very good magic population developed, then definitely go for the NGS approach for which is a resequencing approach that will give you a much better result as compared to SNP chip. Now there are disadvantages and advantages. Like say for example, in case of if you go for a resequencing approach, but if you do not generate a good coverage data, that means say like for example in case of JK population that we are targeting something like 25x coverage. Okay, so but if you do not generate a good data, then there will be a lot of missing SNPs, and that missing SNPs can be a problematic actually, but in case that is the major advantage with cotton SNP chip, because cotton SNP chip these are probe based genotyping, so there you don't have that much missing data. So each of this technology has advantages and disadvantages, but definitely if you ask me personally, then definitely I will suggest you to go for a NGS based approach than a chip based approach. Okay.
Yeah, thank you, sir.
Yes.
Yeah, anybody else?
Yes, sir, there is Chandrasekhar Patil.
Patil.
Two persons have raised hand.
Hello, sir.
Yeah, Dr.
Yeah, please.
Yeah, Dr. Patil.
Thank you for the informative session, sir. This is Chandrasekhar Patil from Seedworks International Private Limited. Sir, you spoke about novel male sterility fertility restoration system. So, how can it be use it commercially? Like, is it a commercially viable or so?
Yeah, it's a very important question. So again, this is the transgenic technology. Okay, so where actually over the years we develop a reversible system actually. So what we are doing that there is a gene called Becklin gene where we have a NBR as a patent. Like say the tapetum degeneration in case of plants is finely tuned actually. It is get activated. During the pollen development at specific stages of pollen development. So if there is any disturbance at that particular stage, the pollen become sterile, because the pollen do not develop normally. So this is the idea which we use to disturb this programmed cell death. So generally the autophagy and programmed cell death, they are in a way inversely proportional to each other. So if you actually induce this autophagy, programmed cell death gets suppressed, so that is the concept which we use. So we overexpress Becklin gene exactly at the time when the programmed cell death happens in case of tapetum degeneration. So when we overexpress that actually at that time, then the tapetum degeneration do not happen and tapetum remain intact, so the developing pollen do not get nutrients and they eventually becomes abnormal. Now, how to control expression of a Becklin gene? So for that, we have developed another systems actually. So where we introduced a mutation TGTA in a Tata box. A Tata box is a very important cis regulatory element of Tata containing promoters. So if you introduce this particular mutation, we have previously shown in one of our publications that if you introduce this particular mutation in the promoter, it completely abolishes the expression of a gene actually, So now interestingly there is another, so like this Tata is been bound by a factor called Tata binding protein. Okay, now there are another mutant which is available in Tata binding protein. It's called TBP mutant 3, which are the 3 different mutations, and because of this 3 mutation it can bind to this TGTA and instead of Tata box and restore the expression from this mutated promoter. So like say for example in what we are doing in male sterile lines actually, or the female lines, we are actually using this expression system. But now that system will not be reversible. So now we are another my student actually, Surendra Pratap Singh actually, So he developed another idea like, if we can fuse it, this TBP mutant 3 with another factor actually, which is called HFR transcription factor. Now HFR and COP1 is a very interesting interaction which actually involved in plant light regulations. Okay, so what we do that we fuse this HFR with TBP mutant 3 and then target this particular protein with a COP1 actually. So we express COP1 in a male line and this particular thing in a female line. So female lines will always have an expression of this Becklin one, but if we cross it now in case of with male line, what will happen that the expression will be restored because there would be the fertility will be restored because the Becklin one, which is actually responsible for this sterility, will not express in case of F1 because its expression will get blocked because of the COP. So this is the system which we developed. We published also. I think this is available. This publication is available. I think you can read this publication, or you can always contact me on my email ID, which is available on my NBRL website. I will be happy to discuss it further actually, with your company.
Thank you, sir.
Nitin Chaudhary, please. Nitin Chaudhary, he has raised the hand. Nitin Chaudhary. Otherwise, I think Tikku sir would like to.
No, I just wanted to add on this male sterility thing. It's a transgenic technology, so that means we have to wait for it to be available for use, right? We don't know when government will approve.
Yeah, actually, for cotton, it should not be. Yeah, I understand that we keep on discussing among ourselves. With you, with Dr. Khadi about transgenic technology, futures of transgenic technologies in India. But at least in case of cotton, I see that transgenic technology still actually should not be a problem. That is why we are still going ahead with transgenic technologies in case of cotton. Definitely, we are also, the further innovation is required that how can we convert this technology into using genome editing actually. So that is actually now presently we are debating the same concept can be made, using a genome editing, which will have a lesser regulatory, constraint as compared to transgenic. So that we are debating presently. So definitely your point is valid, but what we have shown now is a very good technology with a very good male lines and very good female lines. Now our interest is to backcross this into some, say like, we will be taking some of the popular hybrids of Tiara, their male and female parent, backcross it and try to transfer this technology Tiara's.
We would love to do that. So I think at the government level, you all have to push the.
I think that is
If you
going
look
on.
at today's news,
Absolutely,
bears have got the permission now. I think government is for the, I think for the technology now, because bears got the permission and they are going for the sowing this season. So I think let us hope that the transgenic technology is going to come again and people can utilize. Thank you.
Great. Thank you.
there is another Manoj Falak. Manoj Falak, I think he has also raised.
Manoj Palak.
Yeah, very good presentation, doctor. My question is, in cotton, both genetic male sterility and CGMS system is available, and it is widely been used previously, and right now also it is been used. So, what is the advantage of this transgenic sterility system over the existing natural male sterility systems?
Yeah, it's a good question. I think the same question you need to ask to probably there are companies. What I understand that the genetic male sterility system, which is available from Monsanto, it is available with some kind of a fee, and that is not available to every company actually. So that is what I understand from my collaborators actually. So definitely, if we develop some endogenous technology which is freely available to our Indian company, there will be alternate technology which will be available. So I don't know about CGMS, which you said is also available to companies, and which are those companies which are, but I know that GMS is widely being used by different companies, but they are available with a license with Monsanto. That is what I.
No, sir, it is available with every of the company, and it is free of the charge. It is a natural available systems, and it is nothing to do with Monsanto.
Maybe Dr. Tiku or Dr. Khadi can answer.
No, that is true actually. Male sterility is available to everybody, but only thing, with the CGMS system, the general perception is that there is a yield penalty. I don't know whether that still stands, but for GMS is available to all. I don't think, but I don't know what would be the advantage of your system versus that, unless that study is done. How would we? Dr. Khadi is a better judge.
No. There is a yield drag. There is a yield drag in case of.
Khadi sir, you have login from the two device, and that's the reason they will
unmute here. No? Unmute problem is there.
You can unmute only one.
No, that.
So two devices are there. That's the reason it's.
You are on phone and on your computer. One of them you have to mute.
Okay, I think there is a yield drag in case of GMS as well as CGMS system.
Yeah.
That problem is there because, but Dr. Samir, what the technology has come out, I think it will not be there because of the CGMS system is having the diploid cytoplasm of, the D genome. But in case of GMS, again, there are some factors which are affecting the yield drag to some extent. But to the maximum extent of yield drag is there in case of CGMS system where the cytoplasm is coming in the way. Yeah. I hope the system which Dr. Samir is proposing is not going to have this, and it is going to be as good as a conventional hybrid.
Yeah, and it's always good to have another system.
Yeah, and then always good to have multiple approach. So we do not know that which one will work better for which kind of system.
Yeah. Okay.
So
I
it's
think,
already 2:30. I think Khadi sir, you can close the session.
yeah. I think now we are coming to the end of the webinar. Dr. Samir could able to give us a lot of information, and a lot of things have been done at NBRI. I think now the cotton breeders have to utilize these information and come out, because if you take anything, maybe resistance to the sucking pests or bollworms or even drought tolerance,
Yeah.
or even the maize sterility system. Many things have been done through the molecular.
Sir, I have one question, sir, for sir.
Yeah, later. I think you can put
Sir,
in the chat
whatever
box.
the
Chat box you can put in, and we'll answer later. Okay, it is already
Okay, sir,
time now. I think we'll close. So I think it is time now, to thank Dr. Samir for having given us good information, and a lot of things are there with NBRI. I think people should go ahead with the collaboration and utilize the technology and the information developed. And already I have made a mention about the requirement of the country has to be doubled or 2.5 times by 2050. I think it is only possible by utilizing the technologies developed. That too, NBRI is kind enough to help everybody. I think we should utilize and come out with the the yield increase as well as the quality, and meet the requirement of the country. I thank Dr. Samir, and also I thank the organizers for giving me an opportunity to moderate this webinar, and had a nice information. I think everybody has enjoyed. Almost, I think 100 was there almost all the time, and even it crossed 105. That I could able to observe, and it was a well attended webinar, what I feel. Thank you, Dr. Aparna, Dr. Samir.
Thank you.
Thank you very much.
Thank you, everybody. And before closing the session, I just want to inform you all that next month webinar we don't have it because we have the conference, Fiber Farming Conference, which is scheduled 10, 11, 12 in Bhubaneswar, Odisha. And you all are welcome to attend the conference. We have, eminent speaker who will be talking a diverse range of topic. So that's going to be very interesting, and you all are welcome to attend the conference. So see you all in the webinar in April using the same platform with the new speaker.
See you all in Bhubaneswar, Aparna.
Yes, see you all in the Bhubaneswar.
Yeah, that is what. Thank you.
Okay, thank
Have
you.
a nice evening. Have a great day.